So I gave a couple of lectures, and those lectures were about how we think about invasive species. And I tried to convince you there were two numbers that were very important for an invasion. One is the growth rate when it's introduced locally, and the other is the spreading speed after it's established and moves into a new environment. And perhaps by just focusing on these simple numbers, we could understand how we might control invasives, okay? And so the lab today is a little bit different than the lab we did before. You're going to be able to see it in a folder on your desktop. But I'm just going to give a little bit of an introduction by way of a lecture, which just kind of reviews a little bit of what we were talking about last time. And OK, so we were interested, and you can see this on your desktop, hopefully, as well as up here. OK, so we were interested in looking at the dynamics of some kind of species with stage structure. And we could write it down as a matrix equation. And then the idea is that if you have a matrix A, you can calculate the eigenvalue and use this to estimate the population growth rate. Okay, so there's a package in R that allows you to do this very easily, okay? And we're going to use that. And the eigenvector associated with the eigenvalue is what we call the stable stage distribution. And that is the proportion of the different components of the population as the population starts to grow at rate lambda. And then the other thing we talked about was this thing called elasticity analysis, where you perturb the eigenvalue by changing some of the entries to A. And something that's highly elastic is something where a small amount of change in one of the parameters makes a large change in lambda. So if you want to bring lambda down, you want to focus on the entry with the highest elasticity. And so we're going to also do this for Scentless Chamomile. We'll just calculate the elasticity. And these things are very easy to do in R. The other thing that we talked about was saying, OK, let's not use the eigenvalue, but let's use this thing we call the net reproductive rate. It's a bit like R0. And to do that, we need to break up our matrix into two parts. One is transitions that describe survival moving from one class to the next. And the other is fecundities, or the production of new individuals. And that's what we see here, transitions and fecundities. And the idea was we can create this matrix Q here, fi minus t to the minus 1. And if we take the largest eigenvalue of that, that's called our net reproductive rate. Okay, so we'll do this in the lab as well. Okay? And then finally, we talked about, OK, suppose we have this net reproductive rate, and we're looking at applying a biocontrol to try to bring down R0 to a value that's less than 1. We could, say, use an insect pest, such as a seed weevil. And that would affect certain entries in R0. These are the fecundity entries that were in F on the previous page. And so we'll try to calculate how effective this seed weevil would need to be to bring R0 to a value less than 1. And that'll be part of the lab. Okay? Now, OK, here comes the fun part. When you add space, you really have a matrix model with dispersal. OK? And so this was the integrodifference model structure that we looked at last time. A is the same matrix that we saw before. And now, this column comes from the production of new seeds. This is a flower, produces a seed that produces a new flower. This one is a flower that produces a seed that then turns into a low-growing rosette. And then this one is a flower that produces a seed that just stays in the ground as a seed in the seed bank. These are these 3 entries. And so each of these is going to move some distance from the parent plant, and that's dispersal. And so when we think about the dispersal for each of these entries, these 3 have some kind of dispersal function. And I guess this should be, each of these arguments should be y minus x. So there's a little mistake in these slides, okay? But if you're, for example, in this category, this is a seed that's in the seed bank on the ground, and the next year you're still a seed in the seed bank on the ground. So how far did you move? You didn't move, right? You just were exactly still. And so that's what this delta function says. You're not moving. And these other guys that are in the transitions don't move either. And so this little circle here just means component-wise multiplication. And so it's not exactly like matrix multiplication. It's just you put the A11 times the delta there. Say A13 times that thing there. So it's like you would want to do matrix algebra if you didn't know how to do matrix algebra. Okay. It's got a name called the Hadamard product, but it's just, that's what it is. Okay. So this is our model. And it's like the integrodifference equation, but now it has all that stage structure. So you might say, OK, this is kind of like bringing the matrix structure and this dispersal structure together into a single model. And of course, if things don't move, then it's just like a matrix model. And then the question is, how quickly will this invade into a new environment? And then the formula for that is, before I go through the formula, how would you measure seed dispersal? Here it is for this Scentless chamomile plant. The plant grows up, it produces flowers, they produce these pods, okay? And then the seeds from the pods disperse. And so you need seed traps and have one or more plants in the middle of your plot. And then you measure how many fall into the seed traps at different distances. And then from that, you can get a dispersal function. And that's the one that we're treating to be exponential. Okay? And then the formula for this speed, this should be, this is the A here. This is the K. What we need to do is take the moment generating functions of this matrix and then take the Hadamard product with A and then do this calculation. Okay. So I step you through those steps in the lab, okay, step by step. So it's pretty much the same as what we were doing before. It just looks more complicated. Okay, and so that's basically the overview for the lab. And the idea is that these things we talked about in class, we're just going to actually implement in R. And you might be surprised. It sounds like a lot, but I think it's less than 20 lines of R. And I give you all the lines, okay, in one file. But I'm hoping that I'm hoping I know how to get out of here. Yeah. There should be a folder, if I can get to it. You see. No. Yeah, I think I need to go find Jan. Sorry? Yeah, I just want to close. I'm not a Ubuntu expert, unfortunately, although I probably should be. There we go. Okay, thank you. So on your desk top, you'll see a folder called Lewis Lab. And in there is the first lecture, the second lecture, and then the lab itself. And the lab, I'll just give you a quick overview of it before we get into it. This is something I'm hoping we can do in pairs. And so I'll set you up in pairs in a moment. But the idea is there's a little introduction to R, some of which you will have already seen the other day. So you can probably skip a lot of this or work through it if you feel like you'd like to. Then there's a little introduction to vectors, and you could just type this in and see what these commands do. And then there's a little introduction to matrices, because we'll be using those. And so then comes the main part of the lab. And there's 2 packages to install. And the commands are given here. One is popbio. Popbio is a really useful package. It'll do things like compute net reproductive rates or eigenvalues or eigenvectors just in a single command. So it makes it very easy to do these things. And the other is a matrix calculation one, which is right here. And then what we do is we walk through just what we talked about, step by step. And so the lab has a little discussion of what we're doing. And then it tells you how to implement commands in R to do the calculations. And so the calculations in R are interspersed with the text. And so you want to be sure you read the text and then do the calculations, because this isn't really about R as much as about understanding what's going on. And so that's basically it. I'm going to definitely need a tutorial on how to do things. But the commands, all the R commands that are used in the tutorial are just given in this little file here. Okay. So if you don't want to type them in, you can just take them, say, from here, and then you can go Control-C and then Control-V, and well, I haven't installed the library yet, so that won't work. But say take this one here, you can just copy them one by one here, right? So then we see A1, see the vector. Okay. That just saves you from having to type. Okay, so I'm going to number people. 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2. You want to be a 2 or no? Okay. 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2. Christiane, are you a 1?
I think no.
Okay, a 1 and a 2. Okay, find somebody of a different number. Okay, and go to one computer. Okay, so work together in pairs. And so you can help each other and discuss it together. Okay? I won't be lecturing, but I'll be here to answer questions. Okay?